indicator. I've forgotten the other word which is good, but we implant it in your body, we infuse it in your body. So we can get it to a blood vessel, when you get injections, or the only one put the drip and the hospital, they put that drip, isn't it? They're introducing fluid into your plasma or into your ECF. Now, instead of infusing that fluid, you infuse the fluid before A. This fluid, we're calling it a dye, we're calling it an indicator. This fluid can go into the ECF alone, it can go into both the ECF and the ICF, depending on the properties of the dye that we're going to infuse. So we're going to have different types of dyes, and we're going to look at the specific types of dyes that can go only in the ECF, the specific types of dyes that can go both in the ECF and in the ICF. And we'll look at the properties of each dye, the indicator we're using. Okay? Now, there is this solution B. Okay? Let's say this solution B, we don't know the volume. Okay? So, solution A, we know the volume, we know the concentration, we know the mass, and we've calculated it together. Now, there is this, so we have left the body again, we're in a beaker. Okay, we've gone back to the beaker. So, we have this beaker here, we have this solution, we already know what it is, and we're calling this solution B. Okay. Now, this solution B, we don't know the volume. We also don't know the concentration. Okay, and then what else is that? The mass. We also don't know the mass. Okay, we don't know anything about this volume, the fluid that is in the beaker. We're calling it volume B. Okay, now what do we do? So come out of the body, we're in a beaker now. Okay, we open this beaker. Okay. Then this fluid comes in here, correct? So this fluid comes in here, it dilutes. Okay? So when you come in here, you have that. In fact, the best example to give, let's say this is sugar. No, the one moment is your mind. And then water is solution is going to do what? It's going to go down. Okay? Now, we want to now calculate the concentration. So this is our concentration A, isn't it? When we put it in water B, we now have volume. The volume of this fluid, okay, is equal to what? Volume A, right? Plus volume B, correct? Yes. Now, we can measure the concentration of the sugar solution using different methods. That's not the purpose of our discussion today. So let's say we measure this new concentration. Okay? So our initial concentration was 0.05 gram per. Okay? And then we dilute it in water such that after we measure our new concentration, let's say for argument's sake, 0.05 grams per ml. We've got seen the dilution there. I hope you see the new zero that has come there, correct? Oh, I forgot what I had to do. So we're following the story, right? We had this sugar solution, we diluted it in water, And then, using whatever means, we find the new concentration. Okay? Is that clear? Okay. So now we have the new concentration. This is the new concentration. Can I go? Okay, keep going up. Yeah, keep going. These are not the slides I want. There should be another one for happen. Okay, keep going. Hurry up, hurry up, hurry up. Oh, okay, I think it's the one. Go back. Again, go back. Okay, that's it. Okay, that's it. Now I have to tell you to direct my slide. Okay, the next slide. Okay, so measurement of body fluid compartments. We are saying that we get a fluid which is according to our sugar solution A, is of non-concentration, is of non-volume, and is of non-mass. Okay? After administration of the fluid, so forget about the compartment, we are using a beaker. So we get this fluid, we put it here, measure the new concentration of this one. Okay, the next slide. So, okay, go back again, sorry. This is why I like controlling my own slide. Okay. Back. Back again. One last back. Back again. It's too fast. Okay, the next just there. Okay. So what point number 3 is saying that we draw a sample and using whatever method we determine the new concentration. So this is the new concentration for B, isn't it? So we have the new concentration. This new concentration is for volume volume A, sorry, volume A and B. Correct? This new concentration, this 0.05, is for solution A and solution B. Is that correct? Because we diluted them. Okay, the next slide. Like I was saying earlier on, currently we use radioactive substances, but since we are in Africa, we're going to learn our own methods. Second slide, please. So this is what I was talking about. You have the beaker there, which is just the water. You have your indicator. So you have indicator mass is equal to the volume times the concentration. We already did that. Indicator mass A is equal to indicator mass B. What do you mean by that? Now let's go to the new mass. The mass of this indicator has not changed, correct? So it remains 0.05 grams. This is the mass of solution A. Are we following? No one is behind up to that point. Now, let's try to find what's the next slide? Don't rush. What's the next one? No, go back. This is why I was supposed to be controlling that one. Next one. What? Eh? What? Oh.
This is the correct time to run over to Canada. Asha, come on. It's a child. But why?
So, how do we calculate the volume of B? Okay, we have, remember, this is your water, your pure water. You added a new solution, so your volume B, you have the indicator mass. The indicator mass hasn't changed. So, you still have your 0.5 grams. Okay, what volume has changed this concentration is the new concentration we have is what? 0.005 grams per mil. So add our 0.005 grams per mil. Okay, this is going to give us our what? Our new volume, because this volume is what diluted our dye that we had added. So someone give us the answer for this. 100, 100 mil. Because your grams are going to cancel. This is going to go, and then you have the 100 mil. So for you to get this concentration, you needed 100 mils of what of this solution to dilute this one. And that's the same principle that we're going to use in measurement of body fluid. You get a mass, you get a dye, okay? You have this dye, you diluted it in your water here. After you dilute it in your water, you get the sample, calculate the concentration because it has diluted. Getting that concentration, we now use that concentration to find the new volume. How do we do that? The dye has been diluted, meaning that the mass of the dye has not changed. It is still the same, 0.5 grams. However, the concentration has changed. There is a new concentration of what? 0.05 grams per mil. So when you divide that, you now get your new volume of this, which is 100 mil. When we go to the body, we use the same principle. What principle do we use? We use a dye and we inject it in here. Let's say it's coming into the plasma. We inject what fluid comes into the plasma. Let's say we're using EMMANUEL. Or let's just call it A. Okay. So we inject someone with a dye. That dye has certain properties. What properties are those? It's only going into the plasma. So this has gone into your plasma. It has mixed with blood. Okay? Plasma or blood. So this is like your volume B. After a while, you allow the plasma to settle down. It concentrates. After, let's say, an hour or two, you get your new concentration. When you get your new concentration, which is this one, or which is the new concentration, which is A plus B, then you use that to plug into the formula to calculate the volume of the new, using the new concentration and the previous mass that we have used. Clear up to that point? Have we left anyone behind? Okay, the people we left behind, who have we left behind? Quickly, now we wind up. Yes. Question. I'll allow one or two questions. The people we left behind, where we left you behind? Yes?
When calculating the volume.
How we are going to now have to go into the cell. But for us to go into the cell, try to understand it using the beta method. Yes?
When calculating the volume, okay, I make it very, very wrong. But when you calculate it using the mass and the new concentration, we should be calculating for the overall volume.
Yeah, and that's what we want.
So I mean, that is the volume.
We're going to now correct it. But first of all, we need to understand the principle behind it. Yeah. The people we've left behind, where have we left you behind? Very important. Okay. Okay, let's just kind of keep quiet so that we move together and move faster because we don't have time. Yes?
Yes, and then people get confused with where getting the mass.
The mass is this same one here. Remember, the mass hasn't changed. We're using the new concentration. The concentration has changed. Why has the concentration changed? Because we've added the new volume, isn't it? But we want to calculate the concentration of this guy.
Yeah.
Can I correct your answer? Okay, any other question? Yes, I'm listening. Please speak up.
Like when you take it in the black bag, and then how would you know if the result is
What?
I think when you are injecting the when making the after injecting the black bag, how can
You simply withdraw and measure. You withdraw and measure. Yes? I hope we are clear on that one because we are about to move on now to the actual cell compartment volume and how we are going to measure them. So, like I mentioned earlier, I'll just briefly go through it again. You have the dye or you have the sucrose solution or any solution. You know the volume, you know the mass in here. And this method sometimes is referred to as the what? The dilution method. Okay? The dilution method. So you get this fluid, you put it in here, and then you just get a simple sample from this fluid, and you calculate the new concentration. Let's say, for example, we have the new concentration. The mass of the dye has remained the same, only that it has been diluted, and we've calculated the new concentration. So you can use the mass of the dye and then we use the new concentration to calculate the new volume that has been made as a result of combination of A and B. And that's what you have here. So you just get your volume, you plug in your previous mass of the dye and the new concentration. And then you're going to get the volume of this fluid. That is in here. How do we apply that to the body? Next slide. So you have a compartment there. That compartment can be an ICF, it can be an ECF, it can be both the ECF and the ICF. Okay? You add a trace amount of A. Okay? So you add a dye, whatever dye it is, whether it's sucrose, it's even there are a lot of types of dyes that we're going to add. Add it to your to your compartment there, and then of course, some of it is going to be lost to the ECF, but that is negligible. You don't account for it. It's negligible loss. And that's one of the properties that we're going to look at about the dyes that we use for the measurement of body fluid. And then you now calculate your new concentration. What is the new concentration? Amount injected over volume of distribution. You swap that formula and you're able to get your new volume, depending on what volume compartment you're going to use. So when you give a certain dyes are only going to penetrate the ECM. Certain dyes are going to penetrate both the ECM and the ICF. And then, based on that measurement, you simply withdraw blood if it's your plasma. You withdraw, you calculate the new concentration. Using the new concentration and the previous mass that you had injected into your body, you calculate the volume of that concentration. So using that principle, we're now going to use it to calculate the volume of the ECF and ICF. So using a dye, okay, I'm going to calculate the volume for the ECF we are going to calculate the volume for your total body water. These two compartments. Okay? And then we are also going to use this to calculate your plasma volume. So these are the three compartments that are going to be measured. The other ones, the ICF and the IST and the ECF, are going to calculate them based on those answers that we are going to find. So these are the ones that we are going to calculate, and then these are the ones we are going to measure. These we are going to use mathematical equations to deduce the next one. What's the next one? Okay, so what is the criteria for selecting a dye? When we select a dye, remember I told you that this dye, if we're measuring the ECF, for example, it's supposed to just come and occupy the ECF alone. Of course, you're going to have trace amounts coming here, but they are negligible, and we're not going to consider them. So when we are giving a dye, it must mix evenly throughout that compartment. Remember, we're using the concentration, the new concentration as a result of the injection of the dye. So it should be able to mix properly, and it should not be able to penetrate other compartments that are not being measured. So if you're measuring the ECF alone, it's supposed to just go into your ECF. If you're measuring your plasma volume alone, it's just supposed to go into your blood. That's all. It should be non-toxic, or of course, it shouldn't cause any damage to your body. It should have even mixing. Why even mixing? Because we are using concentration. How that fluid is going to be diluted into a new compartment or into a new fluid. Of course, it must have no effect of its own in terms of the distribution of water or other substances. What we mean here is that when we administer this fluid, it's supposed to just come and sit in here, and then after a while, it's going to be excreted in your kidneys or your urine. Okay, it shouldn't have an effect on other parts of the body. It must be unchanged during the experiment. Remember, its concentration. We are basically putting a fluid into your body. That fluid gets diluted. So that dilution concentration should remain the same throughout the experiment. Why? Because we are using concentration to calculate the volume. Of course, the material that should be used should be relatively easy to measure. The next slide, please. What are the types of dyes that we use? Remember, I told you that there are three compartments that we measure. We only measure the plasma volume, which is part of the ECF. We measure your ECF volume as a total. Okay? Remember, ECF is made up of what? Interstitial fluid and plasma volume. So there are certain dyes that are just going to go into your plasma. Your plasma is your what? Your blood. There are certain dyes that are going to go both into your interstitial fluid and plasma. That is the second thing there. And then there are certain dyes that are going to go into your total body water. What do we mean by total body water? It means that when we administer this dye, it's going to go into your ECF, your ICF, and your interstitial fluid, all your total body water. So those are the dyes that we use. On plasma, mainly we use Evans blue. Okay, the first one here. Evans blue is the one that we use mainly for measurement of the plasma volume. For your ECF, we usually use insulin and mantle. So at least you should give an example of two dyes that we use to measure each compartment that we want to measure. And then for your total body water, of course we use heavy water, not really. We use antipyrine and aminopyrine. These are just concentrations of amino acids based on how they are prepared. So please know that there are only three compartments we measure. Your total body water, which includes your ICF, ECF, and interstitial fluid, your extracellular fluid volume, and your plasma. And remember at least, or give examples of at least two dyes that we use to measure each compartment. Next slide. So we go back. You go to the total body water. Those are the dyes which were in your that table that we have calculated earlier on. So those are the dyes we use. But I think we use more antipyrine and amalopyrine. Okay, next slide please. Currently, we use radioactive substances. Okay, mostly the technology now has changed. Even at PTS, you have a nuclear department. They're no longer using this. Yeah, so you have uh, radioactive substances. So when we give this dye is supposed to go into your ECF, your ICF, and your interstitial fluid. So it's supposed to mix totally there. When it mixes there, you go back to this principle. You simply measure the concentration of the dye and the new concentration. So after an hour, you calculate the new volume. Someone was saying A plus B. This volume that you give as the dye is very negligible. So sometimes you don't need to become negligible. So you just injecting 10 minutes, 20 minutes, max 50 minutes. Okay, the next one. How do we measure the other spaces? Now, I think there's another slide there. Yeah, okay, the slide that I was waiting on. Okay, that's an example. Now, this example here has an answer.
We can solve it. We still don't understand. It's not it's hard. Simple. So the
Let's
question
see if you can discuss it or I should move on.
we can discuss it together. See violation. So the question.
So, just for the sake of understanding, okay, you have this. This is okay. You have your ECS here, and then you have your ICS here. So, what that example is saying that it is 150 milligrams. So, this is our dye. Okay, this is our solution A. Solution A, we're going to call it what? Sucrose. And we're saying that in terms of the mass, we're giving 150 milligrams. We give 150 milligrams, correct? Okay? Into a 70 kg man. Okay? The plasma sucrose level after mixing. So we give this, this is the sucrose. Then we inject it into your body. The plasma sucrose level after mixing is 0.01 milligram. So this is like our new concentration. Concentration B. And then we 0.01 milligram per mil. And then, and 10 milligram has been excreted or metabolized. Okay. So out of this new volume, 10 milligram has been excreted. The volume of the sucrose solution so what is our new volume? Our new volume is the mass of this guy remains the same, 150 milligrams. Okay, 150 milligrams. The new concentration is 0.1 milligram per mil. And this gives you a new volume, which is the volume A plus volume B. And when you get that, how are we subtracting the 10 milligrams?
Because a certain amount was excreted.
It has been excreted or metabolized, and that's why we are subtracting that. So that was the highlight I was trying to give you. So basically, your body is like that heater. You can dilute into a dye or an indicator in there, and then we use it to calculate your volume, and that's how we calculate your ECF. When you calculate your ECF, okay, let's say calculate your ECF, and then you also calculated your plasma. Your IST therefore can be equals to what? Your IST will be equals to what? Total A. IST equals to ECF minus plasma volume. Have you followed that one? Yes. And therefore, if you calculated your total body water, you calculated your total body water minus, and you have calculated your total ICM. So this is how we calculate these body solutions by using the dilution principle. That bit I was explaining to you is an example of the what? A dilution titration with a known volume. You inject it into a certain compartment and then once it becomes diluted, you use the calculate a new volume. Someone
So sad. You mentioned in the radioactive dyes they use a UTH. There's no side effect that they can bring.
no, they don't have side effects. The reactive substances are very good. Absolutely no side effects. I even use for treatment actually for certain conditions.
That's okay.
Okay. Okay, guys, those who are working out, just kindly walk out quietly so that we can get questions from your colleagues. Yeah?
Then you code, but you did.
Plasma and large veins. Remember the lecture of the it's just that large has the fluid compartment.
You can't mix anatomy and physiology. Ah, is it not? My head is going to support it, so
Large, yeah, large.
at the end of it, we're going to bite down.
Yeah.
Okay. Lecture, you know, most of the
Five, want to eat yourself and maybe the past is just calm. We understand. We're all tired. It's been a long day. Those of you who have class at last out of the ones that go, we are all tired. So if you are very, very tired and you can sustain the class, you just simply walk out and then you leave us to discuss. But we understand that you don't have to reject. Yeah. Yes, you always have to be given the concentration because remember, the concentration you don't know your volume, you only know your mass. So we have to find you always be given the concentration. And you use different methods to get it. And you can use the machine. I think that's the question you asked. Yes? But I thought we're trying to actually get the volume ratio itself. How do you make those ratios? How do those ratios come about? So we're kind of going to the genesis of volume. You need to understand how we are these ratios going about.
no
So
power
you need to
cutting
know that
that
if you collide or plastic. So it's the same thing as saying that measurement of BP, for example, as many MS, you just memorize what it is. But if you interpret it, how do you interpret it? You need to understand it. So we can use ratios calculations, but we should understand what is happening. How do they measure it? How do they come out to grow? Any other question on body fluid compartments? I think you want to ask question. Okay, if there are no questions, we have to look at one last thing before we close the class. I know people are tired, but we have to because I might not be available on Tuesday. So we have to finish up cell organelles. So under 30 minutes, we finish cell organelles and we're free to go and enjoy our soccer. So go to cell organelles.
the other slide. Those ones are for this.
Okay, so please, we are having a class for 30 minutes. If you feel you can't sustain yourself in 30 minutes, kindly go out. So when we start, kind of start fast and we're not display. Okay? I don't like the noise that is coming out. So we'll be starting cell organelles in about 3 minutes or so. If you can sustain yourself in the next 30 minutes or you want to ease yourself, kind of excuse us so that when we start, keep
Yes,
moving
please, my only
forward.
disappointment. Organelles, boy. My virus. Well, refresh the PC. Instead of having Microsoft, you're using WPAs.
It's done, right? It's done.
So physiology, yeah. Organelles. Yeah, I think it's the one. Yeah, it should be. Yeah. Shave it out. I like this. Yeah, jam. Chashan. Yung sa matapa. Control out. Delete. Eh.
Okay, are we
Okay.
settled now? Can we start?
Yeah. We are. We are.
Okay, let's keep going so that we have to minutes and over on this. I think we did the nucleus yesterday. Keep going. Finish up on the nucleus. Keep going. And we were talking about the endoplasmic reticulum, correct? And I
Yes.
think that is where we ended. We made go back to the previous slide. Okay, so we mentioned that the endoplasmic reticulum is connected to the nuclear envelope. And from there, we mentioned that we have two types of endoplasmic reticulum. You have your rough or your granular endoplasmic reticulum. What is the difference with that one? It only has ribosomes. So it has ribosomes on the surface, and that's what makes it granular or your rough endoplasmic reticulum. We also say that we have the smooth endoplasmic reticulum or the granular endoplasmic reticulum. A colleague was asking, what is the difference between the ribosomes of the rough endoplasmic reticulum? I don't know if that question is clear to you. What is the difference in the function of the ribosomes that are on the endoplasmic reticulum and the ones that are free? Remember, look at the last statement there saying that you have some free ribosomes in your cytoplasm. So, someone was asking, what is the difference between the ribosomes you find on the endoplasmic reticulum and the ones that are in your cytoplasm? I think I put that in the notes. You will be able to see the required ribosomes. Next slide, please. Okay, so in terms of the granular uh, endoplasmic reticulum, so these are the ones without the ribosomes. What is their major function? They are a site of lipid and storage synthesis. Okay, so you have storage and synthesis. For mainly, we find these in lipid secreting cells. So these are your fat cells. Huh? The lipid secreting cells. These are your fat cells. This is why we find a high number of granular or endoplasmic reticulum. A very important feature of these guys is detoxification. So drugs are referred to as toxins. Okay, so drugs, toxins, they are degraded. And therefore, we expect this endoplasmic reticulum to be more present in your liver cells as well. Why? Because the liver cells are important in what? Detoxification. Okay. We have a different type of endoplasmic reticulum. We find it in muscle cells. Okay, they call it the sarcoplasmic reticulum. Let's not bother ourselves for now. We're going to tackle it very well in detail when we look at muscle physiology. But in your books, as you read, you're going to find that thing they're calling the sarcoplasmic reticulum. Very important in release of calcium in muscle. So you have your muscle cells, and your muscle cells are also found in your cardiac muscle, which is your smooth muscle. So in case you are going through your slides or whatever, then you find sarcoplasmic reticulum, you should just know that it's important in contraction, in muscle contraction. So skeletal muscle and cardiac muscle. Of course, they are important in glycogen degradation, or what you call gluconeogenesis. You will learn this in biochemistry, but it's important to know that glycogen degradation and gluconeogenesis. Gluconeogenesis is kind of the formation of glucose, how it's formed and how it's stored. You're going to learn that in your biochemistry. So that is very important, especially for your liver cells. Okay. So in the liver, the rough endoplasmic, the granular endoplasmic reticulum is important. Please remember for detoxification. In case you have an MCQ, what function is more important? Not really more important, but what you should look for more is your detoxification. That detoxification capacity is very important. Okay, let's move forward. Yes. Next one, yeah.
Some books say they also store. So can you
They also do what?
store calcium? The storage calcium.
Yeah, and that's why I say that we're going to discuss it in detail when we look at muscle physiology. It stores calcium, yes, but for you to contract your muscles, for me to move the way I'm moving, calcium has to be sequestrated, has to be pushed out from a contraction to occur. So it has more of a sequestration function than a storage function. So, however, we're going to discuss those nitty details when we look at muscle physiology. But you're right, that's good so but main any other question before we move on? Okay, so we have ribosomes. This is another class or type of organelle we find in the cytoplasm. Of course, they are tiny granules. They are composed of RNA. I'm sure you did this in your biology something. Where you have that picture of the subunits, correct? So you have those subunits, and in the eukaryotic cell, which subunit do we have?
Come again.
40 and 60, isn't it? So you have the 40 subunit and you have the 60 subunit. We all know that these guys are important for what? Protein synthesis. Okay, so we have two types, like I mentioned earlier. You have those that are free in your cytoplasm, and then you have those that are attached to your what? Your endoplasmic reticulum, which makes them the granular or your rough endoplasmic reticulum. Okay, in terms of function, what is the function? Someone was trying to differentiate. That's why the difference is. The ribosomes that are in the cytoplasm, okay? Sorry, the one we start with the ones that are in the endoplasmic reticulum. These synthesize transmembrane proteins. Okay, transmembrane proteins. Transmembrane proteins are the ones that are transversing through your what? Your cell membrane. Okay. Of course, proteins that are stored in your Golgi apparatus, your lysosomes, and your endosomes. We look at all these types of organelles in just a few slides. In terms of the free ribosomes, these are important in synthesis of cytoplasmic proteins. So you can kind of think about your endoplasmic reticulum producing proteins that are for the cell membrane and proteins that are for the organelles, other organelles in the body. What other organelles do we have? You have the Golgi apparatus, you have your lysosomes, and you have your endosomes. Your free ribosomes also synthesize proteins that are found in the mitochondria. And we looked at, have we done the mitochondria already? Okay, we'll look at it in, I think, the next slide, which is important for energy production. Okay, next slide. Of course, this is telling us where our ribosomes are made and how they are secreted. Okay, so they are assembled in the nucleus, transported to the cytoplasm. How? You have the endoplasmic reticulum, which is attached to your nuclear envelope or your nuclear pores. So they are synthesized like that, where the large ribosomes are synthesized in the nucleolus. We looked at that when we were looking at the nucleus. The small subunits, which is your secretion, are synthesized in the nucleus itself. Of course, you require your mRNA, your large, your small mRNA on the middle, it binds and then protein synthesis takes place. You people are experts when it comes to how protein synthesis takes place. You know that it takes place from the 5' to the 3' end. Next slide. Okay, so those were your ribosomes for you. Please remember the difference in protein synthesis of the free ribosomes and the ones that are attached to your endoplasmic reticulum. Yeah?
And then my question is, does it mean that if smooth extended into the rock, the free radical have to come to the smooth attached to the smooth that comes right?
I'm not sure if I'm getting your question very correctly. I don't think they are changing. Endoplasmic reticulum is the same. Okay, you only have one type of endoplasmic reticulum. What makes it rough and what makes it smooth is the attachment of what? So endoplasmic reticulum is just endoplasmic reticulum. The only difference is when you have ribosomes that are attached to it, we call it granule. When you have no ribosomes attached to the endoplasmic reticulum, we call it a granule. Of course, you are going to have different types of concentrations of endoplasmic reticulum based on function. I was talking about the liver cells which are important in detoxification. One of the functions of endoplasmic reticulum is what? Detoxification. So if you go to your liver cells, you're going to have a high concentration of subnuclear endoplasmic reticulum. Why? Because you want more of detoxification. So it's not really that there's a difference, it is what is attached to the in terms of the mitochondria, it's referred to as the powerhouse of the cell. Any other question? Yes?
Sir, how do the ribosomes know that they can't exceed a specific amount of protein to synthesize?
Remember that is being controlled by the nucleus. The ribosomes only receive the what? The mRNA. So when the mRNA comes, depending on the length of the mRNA is what is going to code for your proteins. Protein synthesis, do you remember that in first year, how the mRNA comes and then you have different amino acids there coding for different proteins. So the ribosomes are not controlling protein synthesis, no. That is as a result of the nucleus which produces your mRNA. So whatever length of your mRNA comes through to the ribosomes, it will be produced. Is that clear? Any other question? Okay, if there are no questions, we look at the mitochondria. Why are we calling it the powerhouse of the cell? Because of its ability to make energy. Please remember that the concentration of the mitochondria is going to be different dependent on the function of the cells. There are certain cells that require a lot of energy, such as the muscle cells, and therefore they are going to require more mitochondria. How does that happen? It happens through what is referred to as what? Oxidative phosphorylation. Oxidative phosphorylation happens through a process called aerobic respiration. We're not going to go into detail in that. However, when you go into your biochemistry, there is a topic called oxidative phosphorylation, and we're going to see how those enzymes are going to be moving. So as we are learning, try to interconnect these things. Okay, they are connected in one way or the other. So that's oxidative phosphorylation. When you now go into your biochemistry, they're now going to teach you which enzymes are involved, and how it happens. Okay. That is in terms of shape. Of course, we're saying that the sausage shape, they have two membranes. We talked about two organelles that have two membranes. One, we talked about the nucleus. The other one, we talked about the mitochondria. When you come and do your biochemistry, you realize the importance of these two membranes. Okay, it's very important. This space between the two membranes is very important for oxidative phosphorylation. So the enzymes that are found in there, of course, they are lying into your pistate. Did I put a picture of that? No. Okay, we'll take it to the next slide. One of the other most important functions of the mitochondria is what is referred to as what? Apoptosis. We've done the cell cycle, correct?
Yes.
The G1 phase, DNA
Mitosis.
purification. We did
Yes,
that.
we did it.
So, okay, even if we didn't do that, at least we have an idea. I'm not going to ask you questions about that. But what I'm trying to refer to is when you have a problem in your cell cycle, or probably a cell has worn out, it has overworked, it has worn out, or there's a defect in the DNA of that cell, a cell can undergo what is referred to as programmed cell death. Genetically, it's going to die. Okay? And one of the organisms or the organelles that is not organisms, the organelle that is responsible for programmed cell death or apoptosis is your mitochondria. But the first function you should think about is what? Oxidative phosphorylation or energy production. Okay, second one. Next slide. I've already talked about the oxidative pathway, so you don't have to memorize what is the oxidative pathway. That is going to ask you what? Biochemistry. Okay, it's referred to as your oxidative pathway. So this is the pathway which combines your oxygen and your hydrogen and water to form energy. So oxidative pathway, oxidative, that's oxygen. So oxygen is taken into your body. Once you have oxygen, it breaks down water, hydrogen there, and then your energy is going to be produced in the form of ATP. It's not important for us to know that, but you will learn that in your biochemistry very soon. That next bulletin is saying that even if, remember we said that the mitochondria has DNA, but even if this mitochondria has DNA, 99% of the proteins found in the mitochondria are products of nuclear genes, which ribosomes produces proteins that are found in the mitochondria. The ones that are on the ribosomes or the free ribosomes.
The free.
Free ribosomes, isn't it?
Yes.
So that's what that bulletin is telling you. The mitochondria has human DNA. It does have human DNA. It has about 13 proteins that are produced as a result of the DNA contained in the mitochondria. However, even if it has that DNA, most of the proteins, most of the activities that are found in the mitochondria as a result of proteins that are being produced from the nucleus. Biochemistry. Have we done oxidative phosphorylation? So we are going to do, there is a topic called oxidative phosphorylation, and we're going to look at that. Next slide. Now forgive me, I have to start this time because I can't see the bullets that are being projected. Okay. Again, I've already explained this. The number of mitochondria is proportional to the cell types. There are certain cells that work more and therefore they are going to require more energy. And therefore, the number of mitochondria is proportional. So the higher the energy requirements of that cell, the higher the number of the mitochondria. Okay. Those two slides are explaining to you some of the enzymes that are found in your inner cell membrane that are responsible for oxidative phosphorylation. We might ask a bit about them in physiology, but definitely we are going to learn them in your biochemistry. Okay, this is just a brief example of what happens during your oxidative phosphorylation. You have your oxygen coming in, it will bind to certain enzymes on the mitochondria, and those things will allow the protons, which is in the form of hydrogen, to be pumped from the matrix into the intercrystal space. Don't worry about it. I don't even know why I put it. This is biochemistry. They are going to learn it. What I want you to know is energy is produced in the mitochondria using in the form ATP. Okay, ATP. That's what is important. The pathway is not important. What is important is your ATP. Next one. Okay, before that. So these are some of the enzymes that are contained on either side of the membrane. So the matrix is enclosed by the inner membrane and contains enzymes. So remember, your mitochondria has two membranes, one on the outside, one on the inside. Some of the enzymes that are found on your inner membrane are those dehydrogenases. Please don't bother yourself trying to memorize those ones. Chemistry is going to bother you with those things. Okay. The other bullet in there is that mitochondrial DNA is always inherited from the mother. Someone was asking you this question. Remember, in your fertilization, okay, in your fertilization, the sperm, the head penetrates through the oocyte, the tail remains on the outside of the oocyte. Why are people looking at me like they've never done this before?
No, I hate that way. We joke.
Women study the biology.
No. Why you going to do this? Who said?
No, I don't have to. I don't know how to do it. So you have this oocyte here. This is your. Let me not bother. But this is your oocyte here, okay, or your egg, your female. Okay, guys.
Who's that joking?
So when the sperm comes in, remember it has the tail, okay, and then it has the head. So during fertilization, this gets cut off, and the mitochondria is found in the tail. Okay?
So does this egg
So only this goes in. Meaning that in this new oocyte, what you have is only the maternal mitochondria. And that's what we mean by the mitochondria contains only DNA that comes from the maternal side. Okay, next slide.
so this drink we have is from our mother's car?
The other. Okay, any questions on the mitochondria before we move on? Yes? It can be, but I think I was explaining that question to you. You eventually start making your own mitochondria after event. So you're going to learn that. So after event, okay, any other question for the mitochondria before we move? Okay, there's the last question there, yes?
If we're asked to define it, should we talk more about structure or function?
Function. And that's why I was telling you to say, I'm not interested in your enzymes and all those things. What I'm interested in knowing is your function. Okay. But for us to understand function, so why we keep referring to a bit of anatomy is because for us to understand the function, we need to understand a bit of the anatomy. Okay, if there are any other questions, we look at the Golgi apparatus.
Who's that?
Go back to the previous slide.
Oh, making us think that.
Remember we said that it has its own DNA. And because it has its own DNA, I think go back to the other slide. Okay, you check in your slide. We said 99% of proteins in the mitochondria come from the nucleus, and about 1%, because it has its own DNA, therefore it's able to make proteins. Why? Because your DNA, mRNA, protein. Get it? And I think it was in our slide somewhere. That one there.
There is the glucose.
99%.
Transfer DNA.
Okay. Okay, we look at the Golgi apparatus. The Golgi apparatus, I think the picture will look here. What's the next slide? Okay. Okay, so that's how the Golgi apparatus looks like. It looks like a membranous sac. Okay, a membranous sac. Go back to the previous slide. This guy contains vesicles of newly synthesized protein content endoplasmic reticulum. And sometimes people or certain books say that this one is important for transportation of certain proteins that have been produced. And mainly, these are proteins that have been produced from your granular endoplasmic reticulum. Granular, they were just saying that those are the ribosomes in your endoplasmic reticulum. Okay. And if you remember correctly, when we were talking about endoplasmic reticulum, we said that most of these proteins are going to your cell membrane. So these are important for in transportation. So they have transportative properties. Yeah, what like transportation?
No.
Yeah? Okay. So they have that property of transporting proteins that have been produced from the ribosomes on the endoplasmic reticulum, and they are transporting them to different organelles, but mainly they are being transported to your cell membrane. Okay. So the next bullet in there is showing you how they move. They basically move. So you have your proteins that have been produced. They'll be moving as a granule. A granule is just like a ball or something that is contained in it. Okay. So those granules will move through the Golgi apparatus. You saw that diagram we put for you earlier, which is like a sac-like or a membranous structure. So they will move in there, and then when they fuse to when they reach their destination, which is mainly your cell membrane, they will fuse there, and they will leave that Golgi apparatus by exocytosis. Why are we saying that they leave the cell? Remember, we have two types of proteins on the cell membrane. Okay, we have the peripheral proteins and we have the integral proteins. So some of those that are being taken as integral proteins are going to leave the cell by exocytosis. Okay, next slide. Next slide. So this is just giving you an example of what happens in terms of movement. So you have your rough endoplasmic reticulum at the bottom, or your granular endoplasmic reticulum. It has ribosomes. The ribosomes produce proteins. Those proteins are moving as secretory vesicles or vesicles, and then they are moving through the cristae. So those transmembranous structures we saw until they move out like that, either via endocytosis or some of them are absorbed in the cell membrane. Okay, next slide. Next slide, please. In terms of function, apart from transportation, as these proteins are being transported, it is believed, or they tell us to say, these Golgi apparatus are important for post-translational modification. So you have your proteins that have been produced, but they can be modified. Okay, they can be modified. How? They can be glycosylated. This is just addition of sugar molecules. They can have disulfide bonds. Disulfide bonds just help in terms of transportation so that your proteins don't lose their shape or structure. They are also important in folding. Again, this is post-translational modification. So they are just modifying your proteins in relation to the requirement or the function that is required. When we say sorting and directing, as they are transporting, they have those channels that we looked at, that picture we saw. So it's like a membranous structure. So proteins that are going to the cell membrane will be sorted and will be directed in a certain way. The ones that are going to the organelles will also be sorted and directed in a certain way. Okay, the second slide, next slide, please. Oh, okay. That's the Golgi apparatus for us. I hope we have no questions. Do we have any questions on the Golgi apparatus? We
So,
only have two organelles and they're done. In terms of lysosomes, so this is very important actually. There are a lot of clinical scenarios where people don't have certain enzymes in their lysosomes and they have an increase in the number of lysosomes. What are lysosomes? Lysosomes are vesicles. So a vesicle is just like a round structure, okay? It contains fluid inside. In terms of lysosomes, it is made up of enzymes, okay? Enzymes. And these enzymes are responsible for intracellular digestion. So lysosomes are kind of the cleaner of the cell. They clean up certain things. You can have broken fragments of DNA. You can have certain enzymes that are not required by the cell. These lysosomes have enzymes that will eventually break down those different materials in the cell. Okay. Primary, so the two types of lysosomes are based on their maturity. So as the lysosomes are being formed, you have those that have not yet acquired materials or enzymes required for digestion. We call them primary lysosomes. When they mature and they have enzymes that are required for digestion, we call them secondary lysosomes. What do we mean by digestion? Digestion will just mean that this is a vesicle, and then inside your cell there is cell debris. So the cell debris can be in the form of dysfunctional DNA. Okay, ribosomes are dysfunctional. All those cell excreta, so to say, are digested in this structure called the lysosomes. Next slide. So inside this, remember these organelles are membrane bound. So it's a vesicle, it's membrane bound. Inside you have digestive enzymes. Of course, it's acidic. Why is it acidic? Because it's supposed to digest, it's supposed to break down materials. Okay. So you have certain cell fragments. Like I mentioned, you can have organelles such as the mitochondria themselves, the Golgi apparatus. You can even have DNA, so fragments of DNA are found in this lysosomes. Important as well is bacteria. Okay, sometimes certain bacteria are digested by this lysosomes, although very very rare because lysosomes are not part of your immune system. However, there is evidence that you have more lysosomes in cells of the immune system, and it shows you that there's a correlation. Remember, your phagocytes or your white blood cells, they engulf the bacteria, and then you have these lysosomes that are going to eventually break down your bacteria. So sometimes they are referred to as the phagocytic vacuum. Why are we saying that it's a phagocytic vacuum? A phagocytic vacuum is just you endocytosize something, you phagocytosize it like that, and then you swallow it inside. Okay. I think I've explained this slide already. Yes? Yes? No. That's not true. So lysosomes are cleaning up the cell. So think of it as cell, that's why I say cell waste material. And when I talk about DNA, I mean fragments of DNA that are non-functional. Okay, so these are non-functional things. I think the previous slide talked about worn out organelles of bacteria. So it's cleaning up the cell. So how does it do that? Just the debris is waste material. So it's digest the waste material. Okay, next slide. So those are examples of enzymes that you have in your lysosome. Your ribonuclease digests your RNA. So there are certain RNA that are no longer used in a cell. They'll be broken down in this lysosome. Of course, you have the ribonuclease and all those types of enzymes. So this is a very important clinical correlate. Why? Because you have certain people who have no digestive enzymes, and then you're going to find that those patients have a high accumulation of crab-related DNA debris. Why? Because the lysosome has ingested this DNA, but it doesn't have the deoxyribonuclease, so it's not going to digest it. And therefore, that DNA is going to remain stuck to that lysosome. So there's going to be an accumulation of lysosomes in the body, and it can be very serious. Okay, next slide. I think this should be our last organ now. The peroxisomes. Any questions on the lysosomes before we move forward? People are tired now. Okay, this is the last organ that we're looking at, unless otherwise. But again, these are just spherical organelles or vesicles. Okay, they have that diameter. Not interested in that. The peroxisomes are important in transfer of hydrogen ions. Now, your cell is not supposed to be acidic. Your cell is supposed to be maintained at a certain pH. So when you have you have an accumulation of hydrogen ions. Hydrogen ions are going to make your cell to become acidic. So these peroxisomes have different types of peroxisomal enzymes that are going to degrade certain proteins and hydrogens. Instead of making hydrogen ions, they are going to degrade them to hydrogen peroxide and to water and oxygen. So hydrogen peroxide is like, what's the formula for hydrogen peroxide? H?
O2.
This dissociates, isn't it?
Yes.
When this dissociates, what does it form?
Water and hydrogen.
And then you have another molecule for oxygen. So we're saying what is responsible for that breakdown of those peroxidases is this lysosome. Mainly is to prevent acidity of your cell. You want to maintain your pH at a certain level and reduce the accumulation of hydrogen peroxide. Hydrogen peroxide is toxic to the cell. So you require these peroxisomes, break that hydrogen peroxide to water and oxygen. Okay, move on to the next slide. Next slide. We are done now. I already explained what apoptosis is, programmed cell death.